All right, I think we can get started. I'm going to be brief in my introduction because I'm pretty excited to hear the next talk, even though I've heard it before. It's pretty exciting. So I'm Brian Zambrovich. I'm an executive vice president of functional dynamics and chief of velocity and operations at Regeneron. A bit of a tongue twister of a title. I hope to simplify it at some time. But Andre got his Ph.D. in cellular, molecular, and biophysical studies at Columbia. He then went on to do a postdoctoral fellowship at UCSF, San Francisco. And following his postdoc, he joined Regeneron in 2014. And he joined in the I&I group, immunology and inflammation. And he's been working on where IgE comes from and ways for treating severe allergy, which he'll be telling us about today. I think that this is a great example of translational medicine, and he'll be talking about work that he recently published in Science Translational Medicine, and he'll be telling us about where this strategy is as far as clinical development.
Thank you, Brian. And yes, I completely agree with Brian, but I think this is a really nice story showing how we think about science in Regeneron and how like really addressing translational questions can sometimes lead to aha moments where you understand what you need to do therapeutically to address a problem. So without further ado, I'll get started just by introducing the type 2 immune response, which is something that we think a lot about in our group. And really, the type 2 immunity really evolved as a mechanism to deal with parasitic infections. And we've known for quite some time that IL-4 and IL-13 are key drivers of multiple aspects of type 2 immunity. There's an innate component of what IL-4 and 13 does that ranges from mucus production to enhanced barrier function to eosinophil infiltration of tissues. But today we're really going to be focusing on the adaptive arm of the type 2 immune response. Really, this centers around the role of IL-4 and 13 in being able to induce class switching to IgE in T cells. And the reason that's important is because IgE is a unique isotype of immunoglobulin that can bind to the Fc epsilon receptor, which is a receptor that's expressed on mast cells and basophils, which are cells that are loaded with very toxic agents and granules that can be released in order to fight parasites. And so what typically happens is the antigen that binds to the IgE on Fc epsilon receptor cross-links that receptor, and you get degranulation of all of these things like histamine, leukotrienes, prostaglandins. And so you can imagine that this is a pretty efficient system that is desirable if you're trying to expel a worm, but maybe not so great when it gets diverted to respond to innocuous things like peanuts, right? And so this is kind of the crux of why we are here, is because allergies have been increasing in prevalence and severity, especially in the developed world, and we need to better understand how they develop and how the memory to these allergens is maintained in order to intervene. So when I first joined Regeneron almost 10 years ago, the IgE field was actually evolving, and there were many questions that were starting to be proposed in the literature that clearly demonstrated that IgE-switched B cells are unique among B cells in certain aspects. But there were still things that we didn't quite understand. So the first thing that we know is that IgE-switched B cells exit germinal centers prematurely. Germinal centers are the structures in secondary lymphoid organs where affinity maturation to pathogen happens and where productive high affinity antibodies get generated. And several groups clearly demonstrated that the switching to IgE is pretty much incompatible with remaining in the germinal century. The other thing that was becoming pretty clear is that at least in short-term models of immunization or parasite infection, IgE plasma cells were detected in secondary lymphoid organs, but not in the bone marrow in these models. And lastly, one thing that was also becoming pretty clear is that the IgE PCR actually signals not just to plasma cell differentiation, but also to mediators of apoptosis. And really, this is a mechanism by which IgE-switched B cells differentiate into short-lived plasma cells and then die off very quickly. And with very little opportunity to develop either memory B cells or long-lived plasma cells. However, this couldn't possibly be the whole story because we know from clinical observations that there were certain things that were consistent observations with the presence of at least long-lived bone marrow plasma cells. For example, there are many case reports of transfers of allergies during bone marrow transplant, where the recipient of a bone marrow transplant acquires the allergy of the donor. There's also evidence that allergen-specific IgE is maintained in circulation, even in the absence of allergen exposure for years, really. And finally, there was clinical evidence coming both from our own drug, dupilumab, but also other approaches by other companies that were aiming to stop the de novo production to IgE, that showed that these approaches could reduce but not completely eliminate serum IgE. And so on the left here is showing a figure from a clinical trial with pyruzumab, which was a monoclonal antibody that targets membrane IgE on IgE-switched B cells. This antibody led to a reduction of about 30 to 40% of IgEs, but it kind of plateaued at that point. And then lebrikizumab and our own drug, dupilumab, target IL-4/13 in the case of Dupi and IL-13 in the case of lebri. And both of these inhibit class switching to IgE. And what we see is that with lebri, we see about a 20 to 30% reduction by week 32 after treatment. And with dupilumab, even after taking it out to a year, across multiple indications, we see a very similar trend with all of these patient populations where you get about a 70% reduction in IgE levels after 1 year, but then it kind of plateaus there. And if you're severely allergic to especially food allergens, that's not enough to desensitize you to those allergens. So with these observations, we started asking the question of what are really the sources of IgE memory in humans, and can we model this in a mouse? And for this, in our group, we use pretty routinely a house dust mite exposure model. And what we have found is that this model really evolves and changes depending on the duration of house dust mite exposure. So the way that we do this is we expose mice to house dust mite extract 3 times a week intranasally for either 4 weeks or 15 weeks. And so in the 4-week model, we know that this exposure is sufficient to trigger eosinophil infiltration into the lungs. We see increases in type 2 cytokines such as IL-4, IL-5, and IL-13. We start seeing some goblet cell metaplasia with increased mucus production, and we also see modest increases in serum IgE. However, if we take this house dust mite exposure to 15 weeks, the model now evolves into a mixed type 1, type 2 model, similar to what is seen in severe asthmatic patients, with infiltration of the lungs of both eosinophils and neutrophils, mixed type 1 and type 2 cytokine production, persistent and severe airway remodeling, and a robust increase in serum IgE. So we decided to use this to address the question of the sources of IgE in allergy.
Did you have also the granulation of the cells, or in this kind of model?
Yeah, so we do see increased mast cells in the lung. And the question of whether degranulation of the mast cells is playing a role is, we're not entirely sure.
Playing the role, I'm sorry to very playing the role in the the pathology of severe lung allergy or in the model, the mouse model?
Yeah, so we view this as a model of severe asthma, right? And so the work that I'm going to be presenting is solely focused on the adaptive arm of that. And so the reason, I'll get into the reason why we're using this to address these questions. But yeah, I mean, with this introduction, what I want to make sure is clear is that there's a lot more that's happening in that lung that is not just the adaptive system.
Fair enough.
Yeah. So to start proving this question, we made a reporter mouse where we could easily track IgE plasma cells. So the plasma cells are marked with one fluorophore for IgE, another fluorophore for a plasma cell marker, and we can follow them during the phosphosmate exposure. And what you can see is, as I mentioned, at 4 weeks, if you look at serum IgE, you start seeing increases in the levels of circulating IgE, but these increases are much more pronounced by 15 weeks of exposure. And then now if we look at IgE plasma cells in the bone marrow, what you can see is that by 4 weeks, we don't really see many in the bone marrow, but by 15 weeks, we start getting accumulation of a very robust and discernible population of IgE plasma cells. And these are all expressing the plasma cell marker BRM4, none of them express the B cell marker B20, so they are differentiated plasma cells that are secreting IgE. Furthermore, we then took this model to a model where we exposed for 15 weeks and then rested for 9 weeks. And what you can see is that even though you do get a decrease in some of these plasma cells, the plasma cells are still there after you stop HDN exposure. The serum IgE is also largely maintained. And furthermore, if we now follow the exposure over a period of like even 50 weeks or 48 weeks, I think we did here, you can see that after about 30 weeks, the levels plateau and you kind of cap the reduction at that point. So then we did another experiment where we, after the 15 weeks in HDN, we depleted B cells with an anti-CD20 and then followed for 32 weeks of rest. And what we can see is that even after B cell depletion and 8 months of rest, these IgE plasma cells were still detectable in the bone marrow and still maintaining the IgE, arguing that these are really long-lived plasma cells that are there for the duration. And furthermore, we confirmed that this is pathogenic IgE that's being secreted by these cells. So what we've done here is we took the sera from either saline-treated mice or mice treated with HDN for 4 weeks and 15 weeks. We normalized the amount of IgE to 25 nanograms and used this to sensitize the mast cells in the ears of mice, and then followed that with an IP challenge of the allergen, which is durably. It's one of the main allergens in phosphosmate. And we do this in a solution with Evans fluid. And so this goes in circulation, and if the mast cells degranulate, then you get extravasation of this dye, and we can measure this in the tissues. And what you can see here is that the sera from the mouse that were exposed to HDN for 4 weeks did not induce much mast cell degranulation, but by 15 weeks, this sera is much more pathogenic and able to induce degranulation. Furthermore, we were able to source bone marrow plasma cells from allergic donors, and we cultured these bone marrow mononuclear cells for 7 days in a plasma cell supporting media, and then used the IgE secreted into a supernatant to sensitize the ears of mice that were humanized for the FcRn receptor. And what we found is that when we do this with sera from allergic donors, we can induce the mast cell degranulation with this IgE from the bone marrow. And so we did this with the allergic donor here with the PLD1 challenge, PLD1 being the main allergen. And we also did it with an olive allergic patient challenging with OE1, which is the main allergen. So at the end of this, we published this manuscript in 2020 where we showed that chronic allergen exposure could drive the accumulation of these long-lived IgE plasma cells that persist in mice irrespective of allergen re-exposure for at least 8 months, that are sequentially switched via an IgG1 intermediate, which allows them to gain affinity and become anaphylactic. And we showed that these are also present in the bone marrow of human allergic patients. So then
this differ from the general thought about.
Yeah, so I mean, the sources of IgE memory have been hotly contested for decades in the field. And I think that at this point, you know, the field was really focused a lot on the idea that you can maintain allergen memory and as IgG1 memory, but then can switch back to IgE, which is still true and is still the case. But that didn't explain where the circulating IgE is coming from when you're not being really exposed to allergen. So I think that this was our main contribution, and I think the fact that these cells can be long-lived and no longer require de novo generation, it is what highlighted to us that you really need to address this in a therapeutic strategy if you're hoping to completely eliminate allergy.
So what would happen in the bone marrow of patients that are allergic bone marrow? Would that be a genetic IgE type of mechanism that you have?
No, it's acquired. It's acquired through
Through the bone marrow
well, so the bone marrow is the reservoir where long-lived
of precursor, absolutely.
of long-lived plasma cells. So if you get vaccinated to measles, for example, the reason that you still have measles IgG present decades later is because you have bone marrow plasma cells that have seeded there, and that's their survival niche. I think the field for a long time was puzzled by the fact that these were not readily detectable, either because the methods were not there or they weren't looking in the right models. And so I think the contribution of this was really to show that in settings of an allergy model, you find these long-lived plasma cells that secrete IgE. And they no longer require IL-4/13, they no longer require B-cell differentiation. They're just there, they're cranking out a lot of IgE from the bone marrow, and they're supported by the bone marrow environment.
So in bone marrow transfer in irradiated mouse, you would definitely see the development.
Well, you see it in humans, in humans that get bone marrow transplant. Yeah. So, so then, you know, we went back to this question because obviously we have all of this data with diplomat showing that you can get about a significant reduction. But we really started asking, is this plateau really, this residual IgE really being maintained by the bone marrow plasma cells? So we went back to our models and tried to model this.
Why do you think it's so challenging for an anti-IgE antibody treatment?
So, that's a good question, and, you know, there's a long, complicated answer, and there's a short one. The short one is, I think when you switch to IgE, you're expressing IgE on the surface. And if you come in with an anti-IgE that can detect the surface IgE, you may be able to induce apoptosis in those cells. But you have to contend also with the fact that the IgE is going to be loaded on the mast cells and base cells, and you could be triggering degeneration. You find a way of targeting those cells without touching the mast cells and base cells. the IgE, then you still have to contend with the fact that long-lived plasma cells may be downregulating surface IgE in the lung. So you need a mechanism where you can really be sure that you're getting those stuff. And we're thinking about all of those things. So yeah, in these experiments, what we're showing is we're now going for alpha inhibition with the build-up surrogate antibody. And so what we're showing here in the top model is if you start dosing an anti-alpha before you give the plasma and you keep it on board, you can completely prevent the development of IgE-switched cells in the skin. And you can completely prevent the serum HDM-specific IgE from showing up in these mice. And that's seen over here. However, if we now go to a 15-week HDM model and we give HDM for 12 weeks so that we allow enough time for those IgE bone marrow plasma cells to develop and accumulate in the bone marrow, and then we start treating with the anti-alpha, what you can see is a slight reduction in the IgE plasma cells in the bone marrow, but they're still there. And the same applies to the impact on serum HDM IgE. So we get a slight reduction, but it's still there. So we really felt like in order to completely reset the system, we needed to find a way of targeting those plasma cells. And so how do we do that? And so as it happened, as we were doing all of this, our colleagues in oncology had developed this therapeutic drug. It's a bispecific antibody for use in multiple myeloma, which is a cancer of plasma cells. And so what is BCMA? BCMA is a cytokine receptor that binds the APRIL and BAFF cytokines that mediate cell survival. BCMA is very specifically expressed in plasma cells. Its expression was regulated during B cell activation so that it's not present on B cells. It comes on as B cells are differentiated into plasma cells. And so what our colleagues in oncology have done was develop this bispecific antibody that can bind BCMA on plasma cells via the targeting arm, and then paired that with the CD3 binding arm as the effector arm in order to recruit T cells to plasma cells and trigger T cell cytotoxicity that can kill the plasma cells. So with this in mind, the question that we asked was, could we potentially completely eliminate pathogenic allergen-specific IgE by transiently ablating the plasma cells with a BCMA by CD3 antibody, and at the same time prevent the generation of new IgEs with an anti-alpha? So that's what we went ahead and tried to model. And that's before we did that, though, we needed to create a mouse system where we could do this. So we humanized both BCMA and CD3 in the mouse and showed that if we inject the BCMA by CD3 bispecific antibody into these mice until this 1-week later, what you can see is that we get a very nice dose-dependent depletion of the bone marrow plasma cells in these animals. The memory B cells are not impacted, and we pretty much looked at every B cell subset that we could think of, and they were not impacted. So now that we have the system working in mice, we went ahead and did the experiment that we had been really wanting to do. So in this particular experiment, we exposed mice continuously to health dose mice for 19 weeks. At week 12, after we know that they've developed the long-lived plasma cells, we started dosing continuously with an anti-alpha to stop the production of the novel IgE. And then at week 15, we came in with a short pulse of 2 doses of BCMA by CD3, and we started bleeding the mice for 1, 3, and 5 weeks. And then when we look at the levels of serum HDM IgE, the results were really striking, because what we saw is if we dose with just the BCMA by CD3 alone, what you can see is that this is sufficient to largely drop the production of HDM-specific IgE. But then as you lose the exposure to that transient dose of BCMA by CD3, and as you continue dosing the pulse-dose-mate, you rapidly recover the production of the HDM IgE, and by 5 weeks after that transient dosing, you're back to where you were, back to square one. However, if you do that transient dosing of the BCMA, in the presence of continuous alpha blockade, what you can see is that you bottom out this response and you maintain it over time. So really, you're stopping the production from the pre-existing cells with BCMA by CD3. You're stopping the de novo production of new IgE-producing cells, and the combination of those two is completely resetting the system. So then we wanted to see if this approach would really be sufficient to reverse sensitization to allergen-induced anaphylaxis. So for this, we did a similar model. We exposed mice to HDM for 15 weeks. At week 12, we started continuous dosing of IL-4 alpha. At week 15, we did the transient dosing of BCMA by CD3, and we took continuous bleeds throughout a 10-week rest. And then at the end of the 10-week rest, we challenged systemically with IV-DRP1, which is the main allergen in pulse-dose-mate. And the reason for waiting those 10 weeks is that we know that once IgE is down to mast cells, it can last for a very long time. So we wanted to give time for those mast cells to turn over sufficiently. When we do the systemic challenge, we measure temperature drop as a measure of systemic anaphylaxis. And then the data were really striking. So what we see is that both in the control groups as well as in the monotherapy groups, we see a very robust drop in temperature, demonstrating that the single treatment of BCMA or the continuous treatment of IL-4 alpha alone are not sufficient to reset the sensitization to anaphylaxis. By contrast, if you look at the green line, it's really indistinguishable from saline mice that have never seen HDM. So this sort was really, really striking. On the right graph, you see the single animal data for the temperature change at 30 minutes, and you can see really that this green group is indistinguishable from the saline group. Now, we also followed levels of serum HDM IgE in this experiment, and what is interesting is that consistent with what I showed you before, you can see that neither monotherapy group is sufficient to completely eliminate the IgE. The combination group, you completely eliminated, but now in this last time point was taken after the systemic challenge. And what we can see is that even after you did systemically IV, the DRP1 exposure were not triggering regeneration of the serum HDM specific IgE. But when you look at HDM specific IgG, what you see is that this exposure to the IV-DRP1 increases the levels of HDM specific IgG in both the BCMA monotherapy group as well as the combination group. So what this is telling you is that IgG memory is preserved, that whatever impact where the BCMA might be having in the pre-existing IgG population can likely be replenished from the memory reset compartment when you see that antigen event. To further probe into that question of how recall responses are being maintained, we did another model where we exposed for 15 weeks to HDM. We did the combination treatment with the alpha treatment and the BCMA, and then we rested for 9 weeks. But at the end, we did 2 HDM recalls incrementally. And again, what you see in this model is the 2 HDM recalls are enough to give you full recovery of the HDM specific IgE in the BCMA monotherapy group. The combination group is largely suppressed. We had a couple of mice that had lower exposure to IL-4 alpha in this group, and we did detect it, but this is largely suppressed. But the important thing is that the recall response that we saw for HD and IgG is indistinguishable between the BCMA monotherapy and the combo group. So again, suggesting that IgG memory and thus general immunity is preserved. And finally, in this experiment, we also looked at total serum IgA and IgM. We see that the BCMA treatment has a transient reduction, leads to a transient reduction in both the monotherapy and the combination group. But what is interesting is this really is transient, and as the exposure to the BCMA transient dosing goes away, you start recovering the levels of serum IgA and IgM. Then we went into a monkey study, and we did a similar combination treatment. In this case, we had continuous exposure to alpha and single dosing of the BCMA by CD3. What you can see is on the left, we have serum IgE levels. These animals were not sensitized to any allergen in any way. We're just looking at basal IgE in these animals. And what you can see is that the BCMA by CD3 transiently reduced in these animals, and then we saw recovery over time. However, in the combination group with the continuous anti-alpha dosing, what you see is that these levels drop and are maintained. In contrast, if we now look at the serum IgA, what you see is that in both the monotherapy and the combination group, the levels of IgA drop and then they recover over time. And then finally, we did some in vitro experiments with human bone marrow cells. Shown here is the staining for BCMA and the view image of these bone marrow plasma cells. You can very clearly see we detect BCMA expression in plasma cells, but not in any of the B666. And then subsequently, we did a killing assay where we isolate CD8 positive T cells from PBMCs, activate them with CD3 and CD28 coded beads, and then use these activated T cells in combination with the bone marrow targets overnight, followed by staining for plasma cells and B cell subtypes. And what you can see really is when we look at the plasma cell gate, we completely eliminate the plasma cells with the BCMA by CD3 bispecific. This is quantified here over 5 bone marrow donors. And then what we were able to do with this assay is we collected the targets after the killing assay, and we cultured them over time to try and detect IgE-secretion. And so when you do this with a non-allergic donor, you don't see any IgE-secretion in your supernatant. When you do it with allergic donors, you see this nice accumulation over time of the IgE-secretion. But when you take the culture that has been subjected to a BCMA by CD3 killing assay, you see that this killing is able to completely eliminate IgE production. So this really confirmed to us that we would be able to target IgE production in humans. And furthermore,
And both the existing as well as
well, we didn't.
the stimulating.
Yeah, we didn't probe for production of this of de novo IgE in the system. This is just ex vivo with bone marrow plasma cells. But we also have some nice data from multiple myeloma patients that are being included here, where we see that persistent weekly administration of BCMA by CD3 reduced IgE, showing that the IgE producing cells in humans are sensitive to BCMA by CD3. So with this, I'll wrap it up with this model that we now have, which is showing that really we're thinking about 2 different sources of IgE that we need to target in combination in order for this to be a true reset of the allergic immune response. The first one is the pre-existing IgE plasma cells that we think are largely maintained in bone marrow, which are not sensitive to alpha inhibition, but which we can eliminate with the transient dosing of BCMA by CD2. The second part of this comes from the regeneration of de novo IgE from IgG memory B cells. And that's where the continuous exposure of alpha can prevent the switching of those cells to IgE without really impacting the ability to repopulate non-IgE plasma cells from the IgE compartment. So with that, I will wrap it up by thanking everybody on the group who did this work. Cedric and Navneet and Carly are in the audience, Cedric couldn't make it, but they did a lot of the work that you saw here today. And Jamie is an amazing leader. George was heavily involved in all of this, as was not in the meeting. So,
So, I mean, specifically bispecifics in oncology are known cause cytokines and we look at cytokines in these animals treated with BCMA by CD3, and why do you think it's a viable approach for severe allergy?
yeah, so we have done quite a bit of that. What I will say is we've seen a correlation between target burden and how many targets there are in animals and people with the degree of cytokine release, which makes a lot of sense if you think about the number of T cells being engaged, making a difference. And what we have seen is that the plasma cell population in non-cancer model, at least in mice, is much lower than, for example, B cells. So we've seen that the cytokine release is pretty damping. The other part about this is, we now have step-up regimens that prevent the synchronous engagement of the T cells and help mitigate risk in that way. And I think that taking that approach of dose exploration in that way here is not just going to help us mitigate the CRS risk, but we've also built in checks for serum IgE in these patients between doses, so that if we see that the IgE drops before the highest dose, there's no need to continue dosing BCMA, and you can potentially mitigate the collateral damage on other isotypes of plasma cells in that way as well. So I think, yeah.
The good questions. So what happens to the normal human beings?
So I think that, I mean, it's a really good question.
It's like the
We,
split pocket of
we don't have a good grasp of like why humans become allergic. There's a lot of theories out there, but I think that what we have learned about IgE biology and the way that IgE is maintained is that we've evolved the system in a way that makes it really hard for us to get these memory mechanisms in the form of IgE. Right? It's like B cells cannot form IgE memory just because it's incompatible with the signaling from the BCI. And it's really hard to get these cells to accumulate in the brain. So I think that we've evolved like for a reason to avoid generating high affinity IgE responses that can be rapidly reached. I think that for all of the reasons that we speculate that allergies are increasing, we've now co-opted for something like the immune system to other things that we shouldn't be having. And because we're trying to expect to these things, then we're certain that it's going to be with things that wouldn't really be.
So the cancer patients who are given that drug, that antibody, then they're less allergic. Is that a good
Yeah, so
hypothesis?
I mean, from that data, we've had at least a couple of patients that were have had IgE levels that were high enough to be considered allergic. I don't know against what allergens or against what the specificity of the allergy was, but we do see that problem.
The relationship with the multiple myeloma is just fascinating. I would say that it goes for the immediate question. And multiple myeloma, the perspective, I don't know if you see the X-rays.
Yeah, it's very interrupt. I just wanted to emphasize that we're not finding any relationship between multiple myeloma.
So the platform that is common is the plasma cells.
Yes,
And plasmacytosis is the mechanism. I understand. My question to you is, just in continuation of the previous question, do we see in patients having multiple myeloma an expression of IgG and vice versa? Multiple myeloma is a bone cells. I don't know if you saw it, anytime you see the cranium of a multiple myeloma, the diagnosis is just so overt that it's a bone cell issue.
So, yeah, and not in multiple myeloma, but I do know, patients with extramedullary disease, present differently. And, what I will say is, generally, like most cancers, it's a formal or different disease. So people that have either IgE myeloma or IgE myeloma. There
Although
are,
there's a difference, we know it's a variance from IgM and multiple myeloma is an IgG type of.
but there's also IgA. And there have been a couple of cases with IgE myeloma.
Yeah.
We actually have cell lines that we routinely use in the lab for the work that they derived from the IgE myeloma patient. So they're rare that they do that.
Would you see in an allergic patient any bone manifestation? Do we see it with the?
Not really, not. Yeah, especially, there are many different allergic disorders that, we think about when we think about, for example, when the patients were before now. But what we're really talking about here is a very, like I said, the type 2 response is very broad with adaptive components, and here we're really thinking about the adaptive response in the IgE-mediated type, which I think is more relevant in severe acute.
How about in the trial, we only might know whether this works in severe allergy patients?
Right, so we did just start the first clinical trial. It's a small trial, but the last I heard is the first patient was recruited a couple of weeks ago. So hopefully by the end of the year.
So what would happen in your opinion with polarization and mechanism that when you give the small doses in the platform?
That's a very good question. So the way that we think about tolerance is, with things like systemic immunotherapy or immunotherapy, what we're trying to do is dose small doses of the allergen over time and increasing that dose so that you slowly build up your IgG and you switch the ratio of IgG to IgE so that the IgGs can outcompete the IgEs and prevent the allergen from mutating the target. We have approaches in house where we have cocktails of monoclonal antibodies against, for example, LV1 to TAF or LV1 for bridge that we can dose patients and we can achieve the same benefits that we get with immunotherapy with a single dose of immunotherapy. So the real question that we've had here is, if you do the BCMA-DP combo and you're maintaining these patients in DP, and then the patients see their allergen while they're preventing the IgE from coming back, but they're mounting response to the allergen in the form of IgG and secreting a lot of IgG. if we take the patients then off of the duty, is that IgGs or the specific IgGs that they feel going to be protected in that, so that they don't have to be on duty for life and they're just protected. And it's a question that keeps coming up. We don't know the good answer for it. We haven't modeled it. So, but we think that it could potentially be the case.
How long after they miss a week of jobs, how long do you think it will last with them? IgE cells.
I think it would depend on our agenda. But if you go after the, and you listen to for your knowledge and you're going to probably come back. But if you miss a dose and you're working it, then you're probably going to.
And I still want to. Maybe I can put in a plug for the next speaker. The next
No, I
speaker,
lied about who it is.
what's that?
I lied to you about who it was. That was confusing dates.
Oh. I don't want to say.